Hemostasis is the process by which the body stops bleeding from an injured blood vessel. The steps of hemostasis are as follows. 1. Vascular spasm, vasoconstriction. 2. Platelet plug formation. 3. Coagulation, clotting cascade. Vascular spasm, step 1 of hemostasis. When a blood vessel is injured, the nerves in the vessel wall release vasoconstrictors, and the injured endothelium releases endothelin. Both of these cause the contraction of the smooth muscle in the vessel wall. This leads to the constriction of the injured blood vessels, reducing blood flow to the injury site and decreasing blood loss. Triggers of vasospasm. 1. Damaged nerves in the vessel wall release vasoconstrictors, leading to a neurogenic reflex contraction. 2. Injured endothelium releases endothelin, which also causes vasoconstriction. Platelet plug formation, step 2 of hemostasis. The platelet plug consists of platelets adhering to the injury site to form a temporary seal over the breach in the vessel. This plug helps prevent further blood loss until a more stable clot forms via the coagulation cascade. Platelet plug formation involves 3 phases. 1. Platelet adhesion. 2. Platelet activation. 3. Platelet aggregation. Platelet adhesion. When a vessel is injured, the surface endothelium is removed, and the underlying collagen is exposed. The injured endothelium releases von Willebrand factor, VDWF, which attaches to the exposed collagen at one end and platelets at the other. This acts like a bridge between collagen and platelets, allowing platelets to adhere to the collagen. This attachment of platelets to a non-platelet surface, like collagen, is called platelet adhesion. Platelet activation. When platelets attach to collagen via VWF, they are activated through a surface receptor called GP1B. This activation causes a series of changes. 1. Platelets change shape from round to star-shaped, increasing their surface area for attachment to other platelets. 2. Platelets release chemical signals including preformed chemicals stored in granules, e.g. ADP, serotonin, and calcium, which are released quickly. Newly synthesized chemicals, e.g. thromboxane A2, which are released later in the response. ADP causes further platelet activation and aggregation. Serotonin causes vasoconstriction, reducing blood loss. Calcium and thromboxane A2 also contribute to vasoconstriction and platelet activation. Platelet aggregation. Activated platelets release chemical substances like ADP, thromboxane A2, and calcium. These chemicals bind to their receptors and activate other platelets in the bloodstream. The newly activated platelets express another type of surface receptor called GP2B/3A, which allows fibrinogen, a plasma protein, to bind between platelets, creating bridges that link them together. As a result, many platelets bind to each other, forming the platelet plug. This attachment of platelets to each other is called platelet aggregation. As more platelets are activated and aggregated, the platelet plug grows in size and forms a seal over the injury site. Summary of platelet plug formation. 1. When a vessel is injured, underlying collagen is exposed. 2. Platelets attach to collagen via VWF, released by the endothelium. This first platelet attachment to a non-platelet surface is called platelet adhesion. 3. The attached platelets release chemical signals, e.g. ADP, thromboxane A2, which activate nearby platelets in the bloodstream. This is called platelet activation. 4. ADP and thromboxane A2 activate other platelets, and the platelets attach to each other via fibrinogen. This platelet-to-platelet attachment is called platelet aggregation. The platelet plug is temporary and not very stable, as it is primarily held together by platelet-to-platelet interactions through fibrinogen. This is called the primary hemostatic plug. The platelet plug temporarily stops blood loss, but requires further reinforcement through the coagulation cascade to become a stable clot, known as the secondary hemostatic plug. The coagulation cascade strengthens the platelet plug with fibrin strands, just like a wire holding objects together. The last step in hemostasis is the coagulation cascade. The coagulation cascade is the process of converting soluble fibrinogen into insoluble fibrin strands. There are two pathways in the coagulation cascade, the intrinsic pathway and the extrinsic pathway, both of which converge on a common pathway. Each pathway involves a series of clotting factors, proteins in the blood, that are activated in sequence. Clotting factors are proteins produced by the liver and released into the blood. There are 12 clotting factors, numbered from 1 to 13, with the exception of factor 6. The intrinsic pathway is activated when clotting factor 12, also called Hegeman factor, comes into contact with the activated platelet surface on the platelet plug and becomes activated. Activated factor 12 then activates factor 11. Activated factor 11, in turn, activates factor 9. Activated factor 9 activates factor 10. Factor 10, in the presence of factor 5, activates factor 2, also called prothrombin, converting it into its active form, thrombin. Thrombin converts soluble fibrinogen into insoluble fibrin strands. These fibrin strands are cross-linked by factor 13, forming a stable clot. On the other hand, the extrinsic pathway is activated when a tissue factor, also called thromboplastin, is released from injured tissue. This tissue factor activates factor 7 into its active form. Activated factor 7 then activates factor 10, causing the extrinsic pathway to converge with the intrinsic pathway through a common pathway. In both the intrinsic and extrinsic pathways, the final product is fibrin strands. These fibrin strands form a mesh that traps blood cells, such as red blood cells and additional platelets, and strengthens the platelet plug, forming a stable fibrin clot, also known as the secondary hemostatic plug. This process stops bleeding permanently. Thank you for watching. Stay curious and keep learning.